This is TWIS. This Week in Science, episode number 824, recorded on Wednesday, May 12th, 2021. Stop shouting and listen to the science. I am Dr. Kiki, and we're going to fill your head with giant lizards, magneto sharks, and poop. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. The best part of being a human is having a human brain. One that is good at understanding how things work. Yes, there are many other aspects of being human that are full of merit and noteworthy of praise. Art and science, agriculture and cooking, architecture and engineering, woodworking and ceramics, medicine and midwifery, pantomime and puppeteering, all the things we are proud of as humans originate from the seemingly basic ability we have to understand how things work, thanks to our big human brains. Being a human with the ability to learn and understand how things work is such a magnificent superpower that it is constantly amazing us with new wonders of innovation and ingenuity. We have such capacity to think and do, to learn and create, to imagine, to build. With a brain like that, there's nothing humans can't do. There is, of course, that rest of our brain, the ape brain, let's call it. And while it has its uses, understanding how things work is not high on its list of things it cares about, which is why most politicians are successful, why many television shows get watched, and how economic systems work. But with so much attention being paid to entertain the ape brain, you might think the amazing human brain would get bored. And you would be right, because you have an amazing human brain that knows how things work, which is why we offer you this mostly ape brain-free episode of This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back with more science. Another week, another 7 days of fantastic exploration of the world. And yeah, my monkey brain enjoys this and all the science. And I have a lot of great stories for you today. I have stories about poop, but not just any poop, really old poop. I also have a fossil turducken, and yum yum, except it's like an aquatic variety.
Yeah.
And also very old. And I've also got magneto sharks. Justin, what did you bring?
I have a recap of a review of Where Humans Come From, a record for the slowest something ever.
Something? I don't know what. It was really slow.
Yeah, that's
though.
the part that's a mystery. That's the catch. Is the teaser is it's going to be the slowest something, but you won't know what it is until the start. A update on giant lizards, giant marine lizards, I should specify, and one of those negative. I guess it's a COVID story. It's a negative outcome of the being locked down type story.
Yawns. And I have smells.
Of course you brought more yawns.
Yeah.
Oh my goodness.
It's Blair's yawn corner.
I made sure to eat a lot of chocolate before the show tonight, so I'll be set.
No,
With
so
that
you'll
little
be
bit of
crashing
caffeine.
just in time for the animal corner.
No, that's not the plan. Well, as we jump into the show right now, I want to remind you that if you have not yet subscribed to This Week in Science, you can find us on YouTube, on Facebook, on Twitch. Follow us, subscribe, hit the like button if you haven't done that yet, get the notifications. You can do all that so you know when we go live. We are also on most, if not all, of the podcast directories that are out there. Look for This Week in Science, and our website is TWIS.org. All right, ready for some science?
Do it.
Yay! Okay, first story is a pretty big one. Researchers have been working on genetic modification, gene therapy cures for a whole bunch of single nucleotide mutation disorders. And there is a particular immunodeficiency that researchers have been working on for quite a while, trying to use gene therapy to fix adenosine deaminase deficiency. It's ADA. This is also known as ADA SCID, which is immunodeficiency. It is also known as bubble baby disorder.
Oh, yes.
Used to be bubble boy, but it also is found in girls. So instead of worrying about that issue, it's bubble babies. And it's also noticed very, very early, soon after birth, very often, while the baby is still a baby. Anyway, it results in children not being able to exist in normal society because their immune systems really do not function the way that they should. They get all sorts of infections, and any infection can be life-threatening because they have this adenosine deaminase deficiency that leaves them unprotected. Their immune system doesn't work. Well, researchers have been working for years trying to create viral vectors that could introduce the fix into the immune cells, the white blood cells of children with this disorder. However, there have been side effects because of the viral vector that they've been using previously. So several years back, a group of researchers got together and they changed their tact and they started using what's known as a lentivirus. And you may have seen this in the news already with headlines like AIDS virus being used to save bubble babies. It's not the AIDS virus. However, it is in the same family of viruses. It's a retrovirus. And anyway, this lentivirus, they did a clinical trial. It just published in New England Journal of Medicine, a study with 50 children who have been followed for several years. They had a 95% success rate.
Wow.
And when it went bad, it didn't go bad. It just didn't work. And so the two kids that didn't respond to this immune therapy, this gene therapy, they were able to use other treatments like bone marrow transplants or medications to be able to continue to live, just it didn't work the same. However, the kids in which it has worked, they've been following them for about 3 years, and these kids are living like normal kids. They don't have to take antibiotics. They don't have to take drugs to help their immune systems. They don't have to go in for shots weekly to stimulate their immune system to produce cells. They're normal, and it's working and it's lasting. And what they do is they go in and they get stem cells from the kids. They harvest CD38 stem cells and then they transfect them using the lentivirus with the genetic modification. And then they put the cells back in, and the cells get back into the body. The cells don't get rejected, and they're working.
Wow.
Yeah.
That's awesome.
Yeah. We're going to see more and more of this kind of gene therapy to help. It's not a cure necessarily. We don't know how long it's going to last. But they haven't seen any problems so far. Maybe this is the kind of thing that kids need to have a booster shot. Maybe at some point as they grow. But for now, everything is going well. Small group of kids, but then again, this is generally a small population in the first place.
Awesome.
So we're going to have a future without bubble children.
They
Wow.
They were
movies that are made in incredibly bad taste are not going to have any cultural connection anymore.
Yeah, they were all over pop culture for a hot minute in the late 90s.
Yeah, they were. Justin, what do you got? What do you bring? I can't speak tonight. What'd you bring?
Let's see. Okay. Let's start off with the slowest something. We live in a world that is very fascinated with speed, how fast the thing can be. is the very definition of some sports, like a car race, a bike race, boat race. We do this on sled, ski, speed skating, horse racing, dog racing, human racing, all of it, heart racing. Don't fall behind. Got to keep pace. Life is, after all, one big rat race, but some things are meant to be slow. Sloths, they're meant to be slow. That's just how they are. That's just how they roll sometimes. But there's not a lot of quick snails either.
Yeah.
Tortoises, I think, generally speaking, are moving their own pace as well. And then there's also runner Shizu Kanakuri, who finished the 1912 Olympic Marathon in 1967. Having stopped briefly to get married, raise 6 children, 10 grandchildren before crossing the finish line 54 years, 8 months, 6 days, 5 hours, and 32 minutes after he began the race.
Wait, what? They didn't say the race is over? No,
that doesn't count. He has to spend the whole time on the track for that to count.
So what's funny is he spent quite a bit of time. He competed in other Olympics after that, and he had already been a record holder of at least one Olympic race, or at least of one marathon race previous to this. But apparently, the 1912 Olympics had an unseasonably hot spell. I think it's in Sweden. Had this hot snap. One runner died. Only half of the people that started the race even finished it. But there was a thing where if you didn't finish it and you didn't die, apparently, you reported that you hadn't finished it. Shizu apparently got stopped in the middle of the run, got taken in by some farmers because he was so dehydrated from the heat, recovered, and then just went back home and never reported anything. So Sweden had him listed as a missing runner. They just didn't know what happened. And so some years later, they find out, there's this 70-something-year-old man who is the one that was listed as missing. He can technically still finish the race. So,
but he finished in 1967. So
yes.
That's not what your story is about, because that didn't happen this week.
Yeah, exactly.
No, that has nothing to do with this story. That has absolutely nothing to do with this story.
Oh, okay.
But because you asked, I gave you the rest of the story. Of that story, anyway. But the story I was trying to tell is totally different. And that is the Engano Island earthquake, which lasted for 32 years, which is a very long earthquake, you might think.
Yeah.
It's actually the slowest ever recorded earthquake. It culminated in the 1861 Sumatra earthquake, which was apparently a very devastating earthquake in its time. This is according to researchers at Nanyang Technological University, Singapore, NTU. The NTU research team says their study highlights potential missing factors or mismodeling in global earthquake risk assessment, because this wasn't supposed to be a thing, to have a 32-year-long earthquake where things just keep sliding and keep sliding and keep sliding. So these things do exist. They are thought to these slow-moving earthquakes where you don't get a big hit on a Richter scale. You don't feel the tremors and all that sort of thing.
Yeah, because this is my question: what's the difference between a long earthquake and a slow earthquake? Because you said it's 32 years long. That's a long earthquake, but what makes it slow?
That's what makes it slow.
Is it? So this is just this slip sliding event where it's moving along. One plate is moving along another plate. Just slip sliding is just like slip. Oh, I'm gonna wait a little more of a slip. Yeah. It's like if you're trying to balance on a hill and your shoes are slipping down, like eventually you reach the bottom.
Well, every time you have one of those slips, you have a release of energy. And then you would have a recording of that release of energy. What they're saying here is this 32 years just kept moving. There was no tension buildup and catch. It was just kept sliding.
So were they constantly recording some movement then?
What they used, interestingly, was coral formation at the bottom of the sea. The coral just happened to be in the right location for them to see coral where it shouldn't be. And they could see that it was this coral formation that it was crossing, this plate line was crossing, moved at a steady rate of formation from the coral away from it. So basically, what this says too is because they didn't know that this was supposed to be a thing, it means that we may have completely misunderestimated some of the tension buildups and other faults because these silent slippages known to be taking place. Yes, they had been known to be taking place, but over short periods of time, maybe several hours, maybe a month of this activity, but not prolonged decades of moving at paces of a centimeter a month or a centimeter a year, whatever the ranges are. Slowest earthquake ever recorded, 32 years in the making.
32 years.
I don't know. I feel like we need a new word for that. That's not enough.
Yeah. Blair, what's your first story?
Oh my goodness. Well, I had to bring this story because it studies the most Instagrammable bird on the internet, and the winner is definitely one of my favorites. So I agree with Instagram in this case. This is a pair of researchers from the University of Constance and the University of Jena, and they found the most Instagrammable bird. How did they do that? They set up a measurement index. They call this the image aesthetic appeal. So basically just counting likes isn't good enough because some birds are posted more than others. Mm-hmm. So how do you decide which bird is objectively the most engaging in Instagram? So they index normalized numbers of likes across time and reach. They looked at 27,621 pictures of birds that had been posted on Instagram. And after applying their index, they found that the tawny frogmouth is the most Instagrammable bird on the internet.
Really?
Absolutely.
Hadargus strigoides. So here's their thing. So previous research has shown that
Instagram likes
birds
big mouths.
Yes.
Yes.
So that would be my interpretation of this story, but that's not how they interpret it. So that is why I like frogmouths. However, previous researchers looking just at likes on social media found colorful birds to be extremely engaging. But when you kind of normalize it, partially because most people don't even know what a tawny frogmouth is, then you start to see that they far and away are the winner. And they think that's because of where their eyes are. So their eyes are in the front of their face like an owl, but the rest of their head doesn't look like an owl. So it looks like a bird that's like a cartoon that's looking straight at you. And they said that also it doesn't hurt that they often look very mad. And that is a human expression, so you can relate to it. And it's very engaging if you think about social media and getting interactions and things like that.
So what we need to know now is whether the tawny frogmouth really is angry or just misunderstood.
They're not angry. They're just
Does it have resting frogmouth face?
Yeah, yes, resting frogmouth face. They either look mad or just so discombobulated. But anyway,
is it just me? Do they look British? They just look British.
No, they are Australian, so maybe they're
related.
They were sent off to the prison island. But anyway, I think this kind of stuff is it was fun. So I threw it in the quick stories, but it also is important for knowing, okay, if tawny frogmouths are extremely engaging and we need to talk about Australian wildlife and conservation, maybe I'll pick a tawny frogmouth for the front or something like that, right? So it is important to know what animals are the most engaging and how to leverage that to create meaningful engagement. So that's kind of the serious side of these things, but also just the frogmouth one I had to report out.
How can we use one species to lead to engagement with other species?
Yes, indeed.
I like it. I like it a lot. All right. So moving away from the tawny frogmouth, I would now like to take us away from the land of Instagram and Instagrammable moments to the world of poop.
Who says that's not Instagrammable?
There's a world of, there is a world
There,
of
I'm
poop,
sure
isn't
there's a
there?
hashtag for that, but I am not looking it up. Anyway, researchers publishing in Nature this week have analyzed DNA from fossilized poop, coprolites, preserved feces, however you want to say it. They took these human-created rocks that were found in human rock shelters in Utah and Mexico that date back about 1,000 years. They were excavated almost 100 years ago and stored in a museum. So they've been around for a while. Nobody's done anything with them. And a bunch of researchers said, hey, I wonder if we can try and do DNA analysis. They're not so old that the DNA in bacteria that's fossilized would necessarily have completely degraded. So perhaps there's a chance that we can compare ancient human digestive systems with modern human digestive systems and take a look at see which bacteria were in our guts back then compared to which bacteria are in our guts now. The coprolites yielded 181 genomes, according to an article in Science magazine on their news site. They were ancient and they were different, notably because of the fact that they lacked, can you guess what they lacked? Antibiotic resistance.
Oh, I was going to say like lactose.
That's, yeah, I was kind of thinking.
Lactase is what I meant to say. But that's not a bacteria. But that's interesting. Of course they would.
Yeah, it was pre-antibiotic use by medicine, and so people hadn't figured that out yet, and so there were no markers for antibiotic resistance in the bacteria in these guts. In 8 samples from a relatively confined geography and time period, they found 38% novel species, so species that they have never found in human guts before. A Treponema bacteria. It's not known in the industrialized gut microbiome, but they're present in every single one of these old poos. So they think it's not just diet that was shaping things. So if you want to go on the paleo diet, it's not necessarily going to get you back to the gut that our ancestors had.
Okay, can we jump into Dr. Justin's not a real doctor, paleo microbiotics.
That's right. So
Fantastic.
the bacteria
I
were
love
different. It wasn't just because of the food. There were other things at play. They don't necessarily know what the dynamics were. But according to this research, everybody's microbiomes have changed. We have evolved, and so have our microbiomes over the last 1,000 years. We've lost a lot of species of bacteria potentially, but we don't know whether or not our bodies have had time to adapt to the loss of some of these species that we don't see anymore, these novel species that were discovered.
Well, and we also gained, I'm sure, quite a few, and some of the ones we gained might be serving the same purpose as some that we lost, and maybe better reflects our current diet. And there's lots of things that could be going on there, but.
And one interesting note from this paper is that at the time that they were collected, these feces, coprolites, were not considered human remains. However, conversations with tribes in the Southwest led them to understand that those tribes felt that that they were a link to their ancestors, and they were upset that there had been no previous consultation. And so this is the first paleo feces paper that includes an ethics statement. So it's just something to think about as we're looking back in time, especially for those of us who are in the United States and North America, South America, as we're dealing with human history and human remains of any kind. It's human history. And it's a link of some sort. These are conversations that are ongoing, and you can't just take coprolites from a camping site.
That's wild, but also makes sense. But yeah, That's really, it puts an extra layer on the relevance and the implications of that kind of study.
But again, these were taken 100 years ago, so it's good that the scientists doing this study took the time to have these conversations and to include the Native people in the conversation. Do you have another story, Justin?
Oh, I should. This is some important news to anybody who's trying to have babies in the midst of a pandemic. Almost half of women with babies aged 6 months or younger met the threshold for postnatal depression during just the first COVID-19 lockdown. And that is more than double the average rates for Europeans who were studied in this before the pandemic.
But how was it in relation to the rest of people who didn't just have children in the midst of a pandemic? Was it higher than the background rate of depression?
Yes. Absolutely zero people, men or women, had postnatal depression if they didn't have children.
That's not what I said.
But so there's a difference.
And of course there is. But I'm
being snarky about my
poking
response.
A little interesting item here. I'm saying I know they're separate, but also depression in general is peaking during lockdown.
Absolutely. And now here's depression and a screaming 3-month-old baby, and you can't take them out of the house or hand them off to the grandparents or hire a babysitter or
I'm going to say that is a huge part of it. I mean, the social connection after you've had a baby is so important. And when you don't have it, when that support network is gone, that's hard.
There's a story of my mother's friend, Rosie, running with arms outstretched as she got close to the windows at the front of my house. Or as I was an infant baby, newborn baby. For fear that my mother might have gotten to the point where she needed to throw the baby out the window. Didn't happen. But there is times when, especially for a new first-time mother, the stress level of this, plus the hormonal imbalances and everything else, postnatal depression is a very real, very common thing. That you need help. So there's a lot of this reporting. This is one of the things I thought was sort of interesting. The more contact new moms had with people, either remotely or face-to-face, the fewer depressive symptoms they reported, suggesting reduced social contact, as you were saying, during the lockdown may have increased the risk of postnatal depression. However, where women had maintained some face-to-face contact with family members outside of the household, they were actually more likely to have depressive symptoms than women who saw fewer of their relatives.
Oh, weird. Interesting.
Well, yeah, interesting. And then it immediately made me think that, maybe we give better support to friends than we do to family. To family, we might give advice, sure, but we tend to be, I thought of it in reverse. I thought of how my kids halos. You can actually see the glowing halo when they are with their teachers. You can actually, but as soon as they're at home, it's like, parents, I don't have to teach you. The people interact a little different. So I think having that outside of your family contact is an important aspect of that social interaction as well. But yeah, that was a very interesting aspect of this. But this is, the takeaway from the researchers is it really does take a village to raise a child, especially when dealing with all these increased demands and stresses. Some of the reports that were coming out of this are statements from the mothers, first-time mothers saying it's like working without the guidebook. Also, a lot of worry that children were not getting development that they deserved or needed, not getting enough stimulus, that they themselves weren't doing enough. And that they're just exhausted. The exhaustion
is going to be huge. Yeah.
Yes. So if there's any mothers out there, if you know a new mother,
let's just do Mother's Day again.
Yeah.
We need to keep Mother's Day at the forefront throughout the rest of this pandemic, and always the mother. Come on.
They build
Always,
humans.
I have a story that goes back in time a bit. It's marine biology at its heart, but it's a fossil. Again, I guess I really like fossils this week. Researchers published in the Swiss Journal of Paleontology a fossil that had been found, which they call a leftover fall event. And this was a crustacean that looks like a lobster ancestor. Crustacean being eaten by a squid that had just been eaten by a shark. So the fossil that they found, they described it as a belemnite, the belemnite, and the other a crustacean of the genus Proarion. Proarion had a body reminiscent of a lobster with long, thin claws, and the belemnite was in great condition so that they could see that this squid-like creature, the belemnite, had a large part of its body torn away by a shark. You could see the teeth marks from the ripping away of the body of this squid, and the squid had died, fallen to the bottom to the sediments while it was itself tearing apart the Proarion.
So, first of all, really rare to get a good fossil of a squid.
Yes,
They're
but
so
they
soft.
had a great one.
Let
Which is
alone-
unusual. The Proarion, which they said had a body reminiscent of a lobster, that one wasn't as great. And what they think is that it was possibly just shedding its skin, or had just shed its skin, so it hadn't completely, its exoskeleton hadn't completely firmed up at that point.
So it was soft and squishy.
It was soft and squishy as well. Yeah, they observed how these fossils looked and how they appeared to have fallen to the bottom of the sediments, and determined that, yes, indeed, it's like an ancient marine turducken, the crustacean eaten by the squid, eaten by the shark. Very rare to discover such a fossil.
It reminds me of, there's a short movie. I don't know if it was tester footage or just the short film of Jurassic World, where at the very end during the credits, they have this little clip of, that classic South African great white shark throwing a seal up into the air and grabbing it. So it's that. It gets flung up. The shark goes up out of the water, all like, I'm a bad shark. I'm going to eat the seal. Gets the seal. And then this mosasaur, giant marine lizard, comes out and swallows up shark while it's eating the seal.
Yeah.
It happened. I'm sure it happened all the time. While
Here we have evidence.
Yeah, while a predator is after its prey, it's not necessarily checking its tail.
It's like, I'm such a big, bad predator. I'm going to eat this lobster. Ow!
It's been going on for years.
Yeah.
So the shark got a good meal in the end. Blair, you have another story?
Yeah. Speaking of getting bites taken out of you, decapitated flatworms can still sense light.
How?
This is like my third week in a row of invertebrates getting chopped up into pieces. Yeah. This is from the Institute for Stem Cell Science and Regenerative Medicine, the Technology and Research Academy University, and the University of Hyderabad, all in India. Found that flatworms are able to sense light exposure after decapitation. This is looking at planarians, a type of flatworm. They have two eyes, which are connected to nerve cells in their head. They don't have a lot of complex organs, but they do have them. Prior research showed that their eyes can sense ultraviolet light, and when UV light is shown on them, they wriggle away. But in this new piece of research, they cut off their heads, and they continued to sense light and wriggle away without any eyes. So they can survive having decapitation and regrow their heads, as we know. So in the meantime, they can avoid UV damage by sensing the light and moving away. They also found that when the flatworms were in a resting state, their bodies still reacted to light, even in the absence of other sensory information. So that seems like these guys are just hardwired to avoid damage due to UV light, even when sleeping or without a head. So the question is, who else has these weird abilities to sense UV light even when you can't see it?
Yeah, that seems to be the question for the last 3 weeks too. Is like, what else can we chop up?
Yes.
Where are the
tune in next
sensory? Where are the sensory organs? Where is the control system?
Yeah.
What is the light? What is responding to the light to make that action happen? If we had thought it was particularly photosynthetic spots in the head region of the flatworm, but it's not. What else
turns
is
out
it?
you don't need your eyes to see if you're a worm everything that's happening around you. Well, I think that's a legitimate one though. I mean, your skin, the eyes probably aren't that good on a worm to like see. Oh, there's the direction, there's the type. But having a full body sensor that can pick up the UV and be alerting it to which direction to move away from, even probably a lot better system.
This is This Week in Science. Thank you all for joining us. We really do appreciate that you're here with us to learn about science and talk about fun things. We're going to make you more interesting. Can you help make other people more interesting too? That's right. Tell them about TWIS. Help grow our audience. That would be really, really wonderful. Time now for the COVID update. Just a quick one. It's a quick one. The CDC director today wrote, "Today I adopted CDC's Advisory Committee on Immunization Practices recommendation that endorsed the safety and effectiveness of the Pfizer-BioNTech COVID-19 vaccine and its use in 12 through 15-year-old adolescents. CDC now recommends that this vaccine be used among this population and providers may begin vaccinating them right away."
Woo! Sounds like a safer school year next year, at least for middle school and high school students.
Yeah, and although kids tend, like we've talked before, there is a lower infection rate in children. The group of 10 years old and over are known to get more infections than the even younger aged children. 12 to 18 is a very sensitive range, especially in this period of time before this group were being allowed to be vaccinated. Now with the vaccine, it really is going to reduce risk to have vaccines available for this population to be able to reduce disease incidence and severity of disease. This is, it's going to be great. Hope that it makes school a whole lot more normal next year.
Yeah, I mean, and I don't want to sound like a broken record every time we bring this up, but some of the newer variants are affecting children more.
Yeah.
And so this is how we prevent new variants from popping up and using children as little reservoirs. So this is better for the kids and it's better for all of us, even those of us that have been vaccinated.
Yeah. I'd rather not look at my child and go, "Hey, you little reservoir. How are you doing?"
It's a little reservoir.
Cute little reservoir. Yeah, no. Other interesting news. Back in December, a couple of researchers posted on bioRxiv their initial work suggesting that the SARS-CoV-2 virus could integrate itself into the DNA of cells, similar to other retroviral diseases. And the data they presented in the bioRxiv ended up in a Twitter fight and in an online fight between a whole bunch of researchers who said, "You don't have enough evidence. That's terrible science. You haven't done a good job." And it was a big hubbub, especially as vaccines were starting to be rolled out and people in the science community were afraid that this story would provide fodder for individuals trying to promote conspiracy theories about the mRNA vaccines. Fast forward to this week and this new study that has come out in the proceedings of the National Academy of Sciences. They have updated their data. They actually brought in as a collaborator a couple of the biggest critics of their original paper, of their original work. So they had their critics help them make their work stronger. The study is called Reverse Transcribed SARS-CoV-2 RNA can integrate into the genome of cultured human cells and can be expressed in patient-derived tissues. So we don't know what this means. Yeah. Nobody knows what it means that parts of the SARS-CoV-2 virus, not even the whole virus, just parts of it end up in human DNA. Nobody knows whether there are links between that and long-term health effects that we see in COVID patients. But it is thought that this could explain why some patients, even though they don't have any symptoms, why they don't test positive, why they may produce viral RNA after recovery, why on some tests they test positive for viral RNA. This could explain it. But as for the other aspects of implications, nobody has any idea yet.
It's not zombies, though.
Not zombies. And they say that because it's only subgenomic sequences and it's mainly, they say in their abstract, mainly derived from the 3' end of the viral genome, integrated in the DNA of the host cell, infectious virus cannot be produced from the integrated subgenomic SARS-CoV-2 sequences. So this is because they're little segments. They're not the whole genome. They're not going to pop out and be, ta-da, surprise, you're infected again.
And that's the part that will be lost on the majority of media when they pick up this story.
Yeah, no, but it is integrating it. This gets back at a bunch of stories that we've talked about over the last year about how SARS-CoV-2 it enters into the cells and it just wants to make the cells its own. Like it reformats the hard drive.
So it could be zombies. Is what you're saying.
So maybe. I mean, we don't know what these bits of retroviral DNA really want to do now that they're there. I mean, maybe they want to give us superpowers. Maybe this is how we become Superman. I don't know. Yeah.
Use you a sonic cough.
I think I saw that an android in Raised by Wolves. Terrifying.
Oh, yes. Or I guess some sort of superpower where you can't smell anything. Or maybe you're really good at smelling something. I don't know.
I hope, hopefully the opposite. Yeah. But it's interesting. This is just the interesting, unresolved curiosity of certain aspects of this virus and maybe other coronaviruses, other SARS coronaviruses. We don't know everything that this family of viruses does. And so this will give us more information about what it's all about.
I do like the idea of humanity loses its sense of smell, but it makes the other senses that much more intense.
Yes.
Ooh, I can taste everything now. Except that would probably not be true. You'd probably kind of lose your sense of taste.
Yeah, you'd lose your
too, because without the sense of smell.
Yeah,
but then we can hear everything that's going on at the neighbors.
I don't want to hear my neighbors. But that's all I had for the COVID corner, for the COVID update for this week. Just a couple of quick stories. And then it's time at this point to throw it on over to our favorite animal-loving, science-loving host of the show. It's Blair's Animal Corner.
With Blair?
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh, I'm just having trouble paying attention. I'm just yawning. So, so a recent study from Utrecht University. I'm clearing my throat and yawning all at once. It's just really
it's Dutch. I don't think
a finishing school nightmare over
they
here.
have that much in there.
They do when I say it. But anyway, A new study looked at yawning. Once again, we've talked about yawning so much on the show. It's changed over the years on what we think yawning did. Yawning for so long was assumed to function as an oxygenation of blood. But oh my gosh, I'm gonna yawn again.
You started it.
Instead,
This is all your fault.
Recent discoveries by the lab of Gallup show that yawning actually cools the brain, which I do believe we reported on the show pretty recently. So that is the newest theory. Seems to be pretty accurate. There's a bunch of proof to that theory now. There's an inhalation of cool air and the stretching of the muscles surrounding the oral cavities that increases the flow of cooler blood into the brain and has thermoregulatory function. So how they measured that was, for example, they tested the temperature of the brain. Basic, right? The temperature of the brain dropped rapidly after yawning. Ambient temperature determines how often yawning occurs. If you crank up the heat, they yawn more, which I just thought the heat would make me sleepy, and that's why that would make me yawn. But no, it's actually the differential between what temperature your brain wants to run at and the temperature outside, just like the fan kicks on your computer if it gets too hot in the room, right? And they also found that people rarely yawn when you have an ice pack on the back of your head or neck, or you do other things to cool the brain. Also something I've done before when I was falling asleep, but I assumed it worked just because it was kind of shocking to the system. So it would wake you up, jolt you awake, right?
That's what I would think, that the cold would kind of keep you alert.
Yeah, but this is just cooling the base of your brain. Like, this is cooling your spinal
Cooling your
cord.
blood.
Yeah. And so anyway, so all that to say, yawning cools the brain. That's not what this study is about, though. This study is about who yawns more in the animal kingdom and what that means for their smarts, their brain, if there's any connection there. Does a smarter animal yawn more, for instance? So we yawn about 5 to 10 times a day. Sometimes
Gosh, darn it, Blair.
I'm tripling that quota right now. And so there was there you go. There was a strong indication that the duration of yawns are linked to brain size and the number of neurons in the brain. And so that's what this study is all about. This is an international team of scientists, all centered around one biologist, Jorg Madsen of, I'm going to say it again, Utrecht University. And
they don't have it's much more similar to English than you might.
That's how I'm going to say it, though. And Andrew Gallup of the State University of New York Polytechnic Institute. So they wanted to see if the length of a yawn is related to basically how many neurons you have. And they collected more than 1,250 yawns from 55 mammals and 45 bird species. And they did that by visiting zoos. So they just sat around, had their camera on animals, and waited for yawns. And it did take a long time. They probably yawned a lot themselves while they were sitting there waiting for it. But they wanted to see if the size of the animal, if the size of their brain, and the type of animal they are had anything to do with how long their yawns were. They linked the durations of yawns to brain and neuronal data, which was provided by previous studies by Pavel Nemec of the Charles University in Prague. They allowed them to conclude that if you take out body size, the duration of yawning across species increases with the size and number of neurons in the brain in that given species. So you're taking out individual changes, because that's always the kind of the big fallacy that we talk about when we talk about big brains. It doesn't really matter how big your brain is within a species. What matters is how big your brain is on average in your species versus other species. And that's really what matters to intelligence. Yeah, so it doesn't matter if my brain is heavier than yours or yours is heavier than mine. That doesn't really matter. But the fact that your brain is bigger, heavier, and has more neurons than that of a dog, that indicates intelligence. So,
okay.
Yes. So the duration of yawns across species increases with the number of neurons in the brain.
More
Beyond
neurons,
that,
more blood flow, more
Yes.
yawning,
yes,
basically.
it's a bigger fan on a bigger supercomputer. That's all it is. And so mammals also appear to yawn longer than birds. They think that's explained by the higher core temperature in the body of birds. So the differential to it being too hot outside is less. I think it might actually have more to do with the fact of the way that they heat and cool their bodies that we learned about just a couple weeks ago. Is that they heat their body with their blood. So it's a different system.
That might have a lot to do it, actually. Yeah.
Yeah. But so I think that was an interesting connection that I made by looking at the story. But the brain functions overall best at an optimal temperature. Again, just like a computer, right? And so the kind of piece of food for thought that the lead researchers leave us with at the end of this story is that perhaps
Oh God! Oh, you need to stop. Oh gosh.
Perhaps we should stop considering yawning as rude and instead appreciate that what's actually happening is the individual is trying to stay attentive. So basically, this is like, I want to pay attention. This is important. I want my brain at optimal temperature. Let me yawn.
Yeah.
But then we should make yawning be an acceptable thing when you're having a conversation, whereas instead you're like,
yep,
I hate fighting the yawn, trying not to yawn because you don't want somebody to think that you're bored.
Yep.
Yeah.
Yep. But this is a very good indication that it has nothing to do with boredom. It actually has to do with a deep desire to stay attentive.
I need to be attentive. I need to be attentive. It's my show.
Okay, I'm not buying that at all.
Okay.
I mean, I've never been in one of those conversations where it's like, yeah, and then we should do this. Gosh, this is a great idea, or the creative collaboration, or tracking down an idea, or thinking through something, and just everybody start going.
I think you get more sleep than I do, Justin. Or you're used to less. I don't know, but
does that happen? Does your brain overheat and you start yawning because it's such an exciting thing to talk about?
I yawn all the time when I'm in conversations I care about. All the time.
Like right now.
That just doesn't happen. By the way, I apologize to anybody who happened to be listening to the show while
it's making
doing
my
a
eyes
late night
water.
of work or going on a long drive or anything where you're supposed to be staying awake right now. And we keep saying yawn and yawning on the show.
No, but we're making them yawn, which is actually waking them up.
Keeping you awake.
So it's
That's
really waking you up.
We're really helping.
Yeah.
Stay awake.
Yes. Moving on from yawns to smells. I have a story from Francis Crick Institute and University College London. I can't say the word London today. Found that mice can sense extremely fast and subtle changes in odors, and that they can use this to guide their behavior. So this is part of a larger conversation, I think, that we have had before, and that is part of a deep-seated belief that until now has been based on very little evidence. And that is that I think there are subconscious decisions that we are constantly making based on smells. So to that end, that's why I brought this story, obviously, to promote my own agenda. But this study...
We allow you to do that here.
Of course. I'm just going to present the study. I'm going to tell you what I think it means, but it's up to interpretation, baby. That's what this is about. So this study was looking at odor plumes. An example of an odor plume is like walking by as there's steam coming up off of a hot cup of coffee. It's something complex. It's turbulent. It has a meaningful piece of information to it, but it is also brief, and it is dependent on position, and it's not a constant saturation of smell, right? So previously, it has been assumed that mammalian brains can't fully process temporal changes in smell because they're happening so rapidly, which is faster than we can sniff. So if then something is happening faster than I can intake breath intentionally or unintentionally through my nose, if I'm not forcing air into my skull, then how can I respond to these odor plumes that are a fraction of a second long. And so that really is what this gets to, is there's some sort of passive response that's happening that is not related to the forceful movement of air into your nasal turbinate. So they used behavioral experiments where mice were exposed to incredibly short bursts of odor. They used neural imaging. They used electrophysiology in computer models. And they found that mice can, in fact, detect rapid fluctuations with odor plumes at rates that were previously assumed impossible. The mice then use that information to distinguish whether odors are coming from the same or different sources. So they were actually able to behaviorally test them as well, where these odor plumes were coming from, and they were able to respond, even if they were close to each other. And you could see how in the wild it would be extremely helpful to know where a smell is coming from and be able to respond to it. But these really small 40th of a second blips of smell, they're enough to influence mouse behavior. They were able to correctly distinguish between odors that were at this 40th of a second, that's 40 hertz. And the kind of one of the reasons they were specifically looking at this is because of the olfactory bulb. It's, as we've discussed previously, the part of the brain where the nose sends signals to. And the olfactory bulb has a lot of computational power. And so the researchers were looking at this saying, why is this olfactory bulb such a focus? Why is it so large? Why is it prominent? Why is it so powerful if it's being used for occasional sniffs? If it's not directly related to species survival and it's not being used constantly, they would expect it to not be so prominent and important.
I am sensing a bit of anthropomorphism
yes.
in animals. I mean, humans, we think we don't use smell that often. We do.
Mm-hmm.
It's a lot of unconscious responsiveness, I think.
Mm-hmm.
But at the same time, oh my goodness. I mean, to imagine that mice are not using their olfactory bulb for behavioral decisions, that they're not doing massive amounts of processing in the olfactory bulb when they have pheromones. We know they have a vomeronasal organ. We know there is a lot of scent stuff going on. They recognize the scents of their babies, their mothers, their siblings. What?
I think you're totally right here that part of it has to do with the fact that it's hard for us to imagine responding passively or responding actively to passive smells that are constant and tiny and varying. Even though, like I said, I personally think that we do. But yes, because it's something that we're not aware that we're doing, let's say, it's something that hasn't potentially been researched as closely. And so that's one of the cool things that came out of this study is that they actually were able to design new technologies like a high-speed odor delivery device that could measure several odors simultaneously with extremely high precision.
I want them to use this on a person in an MRI.
Yes, please. Yes, please.
Tell me which direction the odor is coming from. Tell me which source. What do you smell right now? Like, why not?
We use pheromones. I'm convinced. We use pheromones. We our memories are often related to smells. We have emotional responses to smells. There's all sorts of stuff going on that, you have smell aversions. Maybe you can't even tell that you can smell that thing that you have an aversion to. And it's so small and so slight, but oh gosh, your stomach doesn't feel good all of a sudden, or you're walking quicker or whatever. But I think there's so much more going on. Like nasal turbinates, man. So our nasal turbinates are pretty small. They're about the size of a postage stamp. But when you unroll nasal turbinates from a dog, they're about the size of a handkerchief. And when you do that same thing with a grizzly bear, it's about the size of a picnic blanket. Are you really telling me all of that, all those cells and all of the nerves involved with those cells are just there for sniffing? I don't think so.
No, it's constantly sampling. I mean, we don't have the moth antennae, or we don't have these noodly appendages like insects have, that we just know they float on the breeze and run into the things in the air, and they follow the plumes in the air because they're running into them, and the chemicals are running into their antennae, and that's changing their behavior. Wouldn't that continue to be constant? That even though we're breathing things in, it's not as direct, but things fly up my nose all the time that I don't want.
Yeah.
But there's also, but you've seen animals do a concerted sniff.
Yes.
Where they're like, this is me checking the air, and there's maybe like even a little bit of a head movement there to maybe it's a directional thing. I don't even know what's happening when an animal, a dog or a bear, is sniffing the air.
Oh, for sure, it's all of it, but
they're really picking up on things that I can't even imagine are present to be picked up on.
And they probably picked up on something passively that is now cueing them to actively investigate, right?
Absolutely.
Yes. And that's exactly what this is kind of getting at. And think about, so people who've reported on losing their sense of smell, we were just talking about this, right? From COVID, and they describe how strange it was. And I'm just thinking about, like, if tomorrow you could no longer see any version of the color red at all.
Okay, so tomorrow I see like Blair.
Right. Okay. So this is my point, is that if you suddenly were missing an entire input, it would be jarring, right? And so I think that's where I'm kind of thinking is there is an entire landscape of smell that is part of our existence that we take for granted. And it's part of our perception of the universe. But we take it for granted because we can't see it.
Yeah.
I
have a friend who can't smell. I'm going to go talk with her after this. So take me around. Tell me about the world.
Yeah.
So this is very largely me. Very few signals get identified coming through as a smell. I can smell almost nothing. I can smell really bad things.
Like cinnamon.
The smell of cat urine and cinnamon. Those things get cut through, and I'm like, oh, avoid, stay away. There's cats somewhere, or there's cinnamon. Christmas is an awful time of year. Because they begin to fake spray. I don't even know if people notice. I notice because it's one of the smells, and I don't like it.
No, it's aggressive.
It's one of the smells I can smell. But they start scenting everything with cinnamon. Department stores, grocery stores, they're like spraying cinnamon on things to get people in the Christmas mood.
But it's good that your sense is, even if you aren't consciously going, look at the nice yummy smells, the things I love. At least it's alerting you to the things that you don't like.
Yeah.
Your warning system works. That's good.
But the idea of this study is that you're not consciously aware, but you are making micro decisions based on micro smells that you don't even realize.
Turns out I might not really dislike that person as much as I think I do. It's just they were chewing cinnamon gum when I met them. And therefore...
Awful person. Terrible person.
I think that is the definition of a terrible person. Somebody who chews cinnamon gum is the definition of a terrible person.
I used to love cinnamon gum.
I used to eat. So this is why I think I have cinnamon aversion. Is my grandmother's counter had the butter that was like soft butter that was left out. I could use the toaster all I wanted, and the bread was out too. And there was a shaker of cinnamon and a little bowl of sugar. And I was allowed to make as much cinnamon bread as I wanted. It was the one thing at like, I don't know, age 6 or 7 or whatever it was, that I could go into the kitchen and just do myself. And I had so much of it, I think, that I think I overdosed on cinnamon at a very young age. to the point where it's just been repulsive ever since.
Maybe
But
you
I ate
accidentally
a
cinnamon
ton.
challenged yourself. And
Yes.
I think I did. I really no.
wanted-
I think that's what happened. I think if a little cinnamon is good and a little sugar is good, more is obviously
It's
better.
great.
And I just kept increasing and increasing, and then probably had a really bad reaction.
We're sorry. We're doing reversion therapy right now, taking you back to your youth. Sorry. This is This Week in Science. Thank you all for joining us again tonight. And if you are interested in supporting This Week in Science, you can head over to twis.org and click on that Patreon link. Choose your level of support and help This Week in Science bring science to more and more people. Your support helps keep us doing this show week in and week out, and we really can't do it without you. So thank you for your support. And now it is the time for Science from Justin.
Oh, is it
What you
that time on the show again? Okay, where did all the humans come from?
Well, first there was a rib, and then
Okay. Let me
No.
clarify. When I
Fish.
asked the
It came
question,
from fish.
where all the humans came from, I implicitly reject, denounce, discredit, openly ridicule any suggestion that the human species resulted from magic words spoken over a lump of dirt, a talking fish, fornicating deities of a cosmic egg origin, or any story that doesn't have a raven in it because it's obviously raven. That one's actually true.
right, of course, no. Where
What
did we
about
come
just fish,
from?
though? Just fish.
So the thing is, right, there's fish, but then there's that's a big jump to billions of humans inhabiting nearly every niche biome on the planet, using up every resource.
Because we're awesome.
Because, well, we're aggressive. We are aggressive.
Pretty much.
I'll give us that. So you'd think that, though, we would have a pretty good idea of where these billions of humans came from, but we have no idea. Well, we have some idea, lots of ideas, actually, which is great because that means that we've been thinking about it and we've been trying to figure it out. But the fact that we don't specifically know is kind of a problem within anthropology, because not having the specific answer means we have a lot of different answers that are coexisting currently of best hypotheses of where humans came from. So, and most of them, pretty much all of them, don't include ravens, which means they're all wrong. But in anthropology departments around the world, the sequence of evolution from ape to human has sort of different discussions going on because we're still in the midst of figuring it out. There's an interesting review titled Fossil Apes and Human Evolution, which is published in the journal Science currently. And it sort of just looks at where we are as far as ape ancestry is concerned. And it comes to the conclusion, current evidence suggests that hominins originated in Africa from an ape ancestor, which may have been unlike any current living species of ape about 6 to 10 million years ago. So that 6 to 10 million years ago is thought to be when we diverged from our closest relative, which is chimpanzees. But what this is bringing up the fact is that there's such a radiation and diversity of ancient ape. There's a lot of apes out there today, but that's nothing compared to the amount of apes that were there previous to the hominin.
I mean, you have something with an opposable thumb. That's how evolution works, right? They're going to try it a million ways and see what sticks.
And it's not necessarily even the opposable thumb, because that's very sort of specific to the hominin line, right? So that's our direct going.
No, I mean, all the great apes have thumbs.
But having it opposable,
yeah,
I thought that was the thing that made it special. Anyway, but the point, so there's that radiation of all the different hominins that we've seen. Yeah, we've talked about that quite a bit. How we're this braided stream of, we got the Neanderthals over here and the Denisovans and the Homo erectus interacting. We got all the hobbit creatures and some of these other things, the red deer cave people, like all these interesting hominins that seem to have been on the planet at the same time, maybe as many as 8 of them on the planet at the same time. So there was a much greater diversity of ape as well in the ape ancestor category, with all sorts of different body morphologies going back through time. There's sort of a gap at around the 10 to 14 million year ago range, where we have very sparse records, which is exactly the period we need to be looking at to understand where the humans come from. Currently, though, what's sort of interesting, some of the closest fits are in sort of Southern Europe, like Greece, or some of them are in Bavaria. Like we have these, sort of more human-looking leg gait in one area, or more human-looking torso somewhere else. You have all these different elements, but nothing that's obviously where the human lineage picked up from. So we may have even been a braided stream before we were hominins. We may have been an ape braided stream before we were even a human braided stream. So it could just be like this keeps happening all the way back, and so it's configuration. So, but some of the interesting things they're saying that our lineage ancestry may never have knuckle walked. Our ape ancestry may not have been tree hangers. We may have already been on the, like, there are so many different variations that we don't know where we came from yet. And that the ape ancestor of not just chimpanzees and man, but maybe gorilla and man, had fewer traits in common with any three of us than what we might expect. That it could be that different because the radiations were so diverse going back through the record. And sideways, having been looking at this, there was, I think I might have closed the thing on it. Oh, here it is. Did you know there was a 10-foot tall orangutan that weighed 600 pounds?
No.
Yeah, isn't that what Christopher Walken played in the newer version of the Jungle Book? No, I'm serious. I'm pretty sure that's what he was supposed to be. He was supposed to be that critter.
It's sort of related to the orangutan. It only died out 100,000 years ago. Gigantopithecus blackie. Distant relative of the orangutan, stood 10 feet tall and weighed up to 600 pounds or 270 kilograms. The picture they have of it looks like Bigfoot. It looks like what you would expect a Bigfoot to look like, exactly like Harry and the Hendersons type Bigfoot looking thing.
So now you're saying there's a chance.
I'm saying there's a chance. But that was only 100,000 years ago. And anyway, the diversity of the ancient ape is a whole other category that we have to go back through to try to find the origin ape. Point of the whole story is it's not going to be linear. Mm-hmm. The whole human and ape history, there's not a whole lot of linear about it.
It's more complicated than we think. We like this nice tree-like, easily branching structure that we can follow, but
it's a bush.
There's a lot more nuance. Yeah.
It's going to be rated stream all the way down.
I love that.
And then there's another radiation of creatures. We're somewhat used to the idea of a creature going extinct because it's a panda, doesn't want to breed. It's a woolly mammoth, and the climate changed on the planet, now it no longer suits them, or they were relentlessly hunted by humans, whatever the case was. There's usually some sort of a decline of a species that occurs ahead of extinction, where they're either being preyed upon or the biome changes, and there's less and less of viable mates. So time takes its toll, and eventually the species goes extinct. Sometimes the extinction can hit when a life form is at its peak, when a species is at the very top of its reproductive killing abilities. Such was the case of a newly discovered version of a giant marine lizard from the end of the Cretaceous period in Morocco, reached up to 8 meters long, which is pretty big. That's like a small school bus.
That's like the size of an anaconda.
It's an anaconda.
Yeah.
Yes.
Yeah.
That alone would be kind of cool, but this is also the third species of this giant lizard to be described from that region in less than a year. And it brings the total number of the giant marine lizards, or the mosasaurs, to 13. Mosasaurs, that's the giant marine lizard that you have seen in the Jurassic World puddle up front, where they were feeding shark stew in the beginning of the movie, and then just shows up to eat things whenever it needed. They were apparently at the height of their diversity and going really strong in the final million years of the Cretaceous period. That's to be going strong for a million years. That's tough. Anyway, with the and then a giant asteroid hit the planet 66 million years ago, and that
was the end of them.
Extinct. Yeah, the newly found species named Pleurodens serpentis had long, slender jaws with over 100 sharp teeth that were fang-like. Used to grab small prey like fish and squid. Fun fact: they didn't eat humans. In fact, there's not a single case of them ever eating a human ever. They had smallish eyes, suggesting poor vision. The snout had dozens of openings for nerves, hinting at the ability to hunt by sensing water movements, change in pressure, that sort of thing. They also had they're distant relatives of sea snakes, and they're thought to use their tongues also to taste the water for molecular clues. And this is one version of it. Some of them had very different teeth for breaking up crustaceans, going after crabby type things. Others went after much larger prey and probably ate other mosasaurs. But very different, diverse beasts. Huge lizards. And they probably acted like them, despite the fact that their bodies very much resembled sort of modern day whales and dolphins. So, diversity of life going backwards is very fascinating. We sort of forget
the diversity of life. You go backwards, you kind of smash species from today together. It's like, lizard dolphins. Yes, of course.
Lizard dolphins. Of course we had to try that.
Crocodile pandas.
Apparently it was very successful. Crocodile panda. That's a thing. You joke, but we're going to find it.
I don't know. Could have been.
Oh,
boy. What, Blair? You mock me? No,
not at all.
She's just picturing a panda with a big crocodile face.
Yeah,
that's right.
I think I was I was looking at pictures of mosasaurs because I wanted to make sure I was correct before I said this. But they do look like dolphins in a lot of ways. But I think they look a lot more like crocodiles in their artist renderings. But I think the thing that's really important to like delineate whales and dolphins from these aquatic reptiles is the way that they move through the water. So the marine mammals go kind of up down, which is why their tail is kind of horizontal. But reptiles in the water, their tail is vertical, and they move more like a snake through the water, left to right. Yeah.
And this is thought to be a relative, closer relative to snakes and Komodo dragon.
Yeah, which the way that I've seen monitor lizards swim and iguanas swim seems like it would be pretty similar to the mechanics of
They've
how
still got that fish-like spinal motion.
Yes.
Very fishy. There's something fishy. Yep. Speaking of fish, let's talk about some sharks.
We have shark stories?
I have a shark story.
Oh, wow.
This one I love because we've been talking recently about navigation, and I was talking about the possibility of the picking up of these magnetic fields in the eyes last week. We had a whole conversation about that. And then this story popped up related to magnetic sense in sharks. Now, we know that sharks have the electric line system that they use electromagnetic fields for sensing their prey, for sensing communication signals in the water, for all sorts of things that being fish living in the water, they make use of all of the sensory aspects that they can. And we also know that several species of sharks undergo very long distance and very precise migrations. Kind of similar to salmon, where they go from place to place. They don't get waylaid. They know exactly where they're going. How do they do it? Do they just look around and go, hey, I recognize this spot. It looks nice. Do they follow the other sharks? What the heck are they doing? Well, a group of researchers publishing in Current Biology this week trapped sharks and then messed with their magnetic sense. They, which of course is what you do. They trapped bonnet sharks, which are known for their migration abilities. They trapped sharks and then took them various places on the planet away from the place that they had been trapped, understanding that by moving them away from where they had been trapped in the ocean, they would be experiencing a difference in the Earth's electromagnetic fields. And then they placed them in a big kind of Faraday cage and then exposed them to different magnetic fields to see how it changed how they moved around and which way they tried to go. And lo and behold, the end of the story is that they were able to show that these magnetic displacement experiments on these wild-caught bonnet heads showed that magnetic map cues are what elicit their homeward orientation, and that it is their magnetic sense that allows them to orient in the correct direction. And this is one of the ways that they are able to get themselves around out there on their migratory routes.
Wow.
Very cool. Well, with an entire structure in their face for electromagnetic fields, as you point out, it would make sense that they use it for more than just finding food.
Yeah, and these bonnet sharks, yes, they similar to kind of hammerhead sharks, where they have this feature of their head that is, they use it for navigation. Their head looks like a bonnet. It's bigger, it's a little more bulbous. They're like baby it's like they're
I think
not
they're very
quite
cute.
hammerheads. They are cute. Look at the big head on that one.
Mm-hmm.
So how is it like? Oh, we got sharks. Maybe the mosasaur had all those electrical sensors on its snout. Apparently, we've got pigeons. We got all sorts of birds that are doing it. We got, why don't we get to have GPS built in?
Maybe you do.
Maybe we do. Yeah.
We just don't pay attention to it anymore. Is that
Or
is
you
that
do
what
and you don't recognize. Like, you know how you talk about how some people have a good sense of direction?
Maybe they're just tuned in to their magnetic sense a little bit better.
Yeah.
Maybe they're tuned in to the Earth's electromagnetic fields. Maybe, though, I don't know, maybe we interrupt those fields too much with all of our electronics. Oh.
Could be.
Yeah, I don't know. I don't think that, like, do we get like a bunch of birds and sharks gumming up the works in the big city just because they got, power transformers and stuff? I don't know.
No, not so much. Yeah,
it's
no.
Different signal.
The pigeons are still able to pigeon.
They're pigeoning just fine.
They're still pigeoning just fine. Yes, but bonnet sharks, they get home using their magnets.
Wow.
Yeah, the homing magnets. And then my final story for tonight involves sticking wireless devices into the brains of mice.
Wait, why? How is it? Oh, it's not wireless at that end. It's just wireless.
It's wireless in the mouse end. So researchers have been trying to use optogenetics, which is a technique in which we can use genetic modification to make neurons stimulable by light. And by shining blue light on neurons, you can make animals do things. You can cause different actions, stop different behaviors. You can make them do all sorts of funny tricks. It's something that researchers hope that we'll be able to do eventually for human behavior. But in the meantime, it's giving us a lot of insight into how brains work. And when we're using this technology in mice, currently it involves genetically modifying the mice so that their brain can be stimulated by blue light, and then sticking a cap on their head with wires that go down and a light that shines in these optic fibers down into the brain to turn the neurons on. But then, because this is electrically stimulated and activated, there are wires coming out of the top of the mouse or rat's head that are attached to a cable that goes to a computer. And if you have a couple of mice that are trying to interact, or even a group of mice, suddenly you have mice tripping over wires and getting tangled up in each other. And so it makes real behavior very difficult to monitor.
Yeah.
Yeah. So you're not really measuring real animal behavior. You're measuring this weird behavior of mice and rats with these things attached to their heads and getting tangled in wires. So researchers wanted to figure out how they could fix that. And so they have created a device, which is wireless technology, which can be like a little hat implanted. It's a little device that can be implanted into the mouse's head. And so the mouse can behave wire-free, battery-free, and it's able to allow groups of animals so that they can examine social hierarchies and the interactions that occur when different behaviors are changed. And so they are investigating how social bonds form and break in animals. And so in their experiment, mostly just a proof of concept, and they will be investigating this more further with this new technology now that they're able to get groups of animals together to do stuff. They're going to turn on and off the neurons that are responsible for social bonding and behavior. Basically, at the flip of a switch, they're telling mice to play with each other, to interact with each other, or to be antisocial, and seeing how that affects group hierarchies and social dynamics.
Play nice with your brother, or I'm gonna flip the switch. I swear.
Swear.
I don't know what's scarier, the idea of cyber mice or the idea that they're playing with the ability to switch on and off mouse interaction. Just used as a model for gosh knows what.
Yeah, so the interesting part of this, it's implantable, and this is the kind of thing that could be implanted. And if there comes a point at which we decide that optogenetics, that kind of genetic modification, is allowable in humans, this would lead to the kind of technology that would control that kind of behavior modifying technology in the human brain. Maybe you need to have something that's not a drug, but that turns neurons on or off to calm somebody who has anger management issues, or to calm anxiety, because anxiety, nothing helps your anxiety, and maybe you need something that can change that. Right now, there's not much we can do except for drugs, but with optogenetics and implantable devices like these, that becomes more of a reality. Right now, it's just studying mouse behavior. It's fine. It's fine, everybody.
It's such a slippery, crazy thing, though, which I always get into trouble bringing it up. But like between human mental health and human control fears, there's that fear.
There is, right? Because it's your brain.
It's a brain. It's human brains.
And individuality is important. In Western culture, it's very important to have your own choices, to be able to choose your own path. Whether or not we actually do is up for debate, but, to have that feeling of choosing our own path is important. Very interesting, huh? Yes, wireless controlled implantable devices. Will this lead to mind control in humans? Story not at 11, because we're not even talking about that. This is just looking at the way that neurons work and the way that brains work to create behaviors in mice. Yeah, the researcher from this study, says they took pairs of mice, and when the mice were near each other, they wirelessly synchronously activated a set of neurons in a brain region related to higher order executive function that increased the frequency and duration of their social interactions. Desynchronizing the simulation promptly decreased social interactions in the same pair of mice. We didn't think this would work, Kozorovitsky said. To our knowledge, this is the first direct evaluation of a major long-standing hypothesis about neural synchrony in social behavior. brains that fire together socialize together. That does it for me, folks. Have we done it?
I think
We've
we
done it.
did.
We made it to the end of another show. Thank you. We made it. All the science that was fit to talk about. Take it with you, everyone. Hold it close. Share it with your friends. Right? Tell people so that they know that the mouse army is coming. This has been This Week in Science. Thank you so much for listening. I do hope that you enjoyed the show. I want to say thank you to Fada for your help with social media and show notes, Identity4 for recording the show, Gord for your help with the chat room, and to Rachel for your amazing assistance. Also, very, 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 very,
That's a lot of varies. That's maybe too many
many
varies.
thanks to
not
our
enough.
Patreon sponsors. Not enough. There aren't enough thanks for our Patreon sponsors. John Ratnaswamy, Kira, Carl Kornfeld, Melanie Stegman, DeKramsta, Karen Tazi, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shubrew, Darwin Hannon, Donald Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Aronovich, Kevin Reardon, Noodles, Jack Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflode, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rabin, Dana Pearson, Richard Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Mark Nazaros, Artyom, Greg Briggs, John Atwood. The profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jerome Depot, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBells, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And for those of you who are interested in supporting us on Patreon, you can find information at TWIS.org. Click on that Patreon link. And on next week's show,
we will be back Wednesday, 8 p.m. Pacific time, 5 a.m. Central European time, Thursday. Broadcasting live from our YouTube and Facebook channels and from twist.org live. And there's a whole list of places where you can find us.
Yeah, so you can watch us live or you can listen to us as a podcast, get some dishes done, fold some laundry. Just search for This Week in Science if our podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
And for more information on anything you've heard here today, show notes, links to stories will be available on our website, www.twist.org. And you can also sign up for a newsletter that's coming out very soon-ish.
Very soon. You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into a lobster that is being eaten by a squid, that is being eaten by a shark, that is also being eaten by a giant marine reptile.
Wow.
Mosasaurus.
You can also find us on the Twitter where we are at Twist Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.